Cardiogram. Principle. The mechanical activities of the frog's heart are recorded by connecting the heart to the starling heart lever and recording using the moving drum. In the frog heart, there are two auricles, that is atria, and a single ventricle. Deoxygenated blood from the frog's body drains into the right auricle through the sinus venosus. The sinus venosus is separated from the auricles by a white concentric line. Start by dissecting the frog to expose the heart. We pit the frog and destroy the spinal cord. We leave the brain intact. Now we lay the pitted frog on its back on the frog board. We pin the frog to the board. We make a median incision on the skin over the sternum of the frog. We enter the chest cavity of the frog and expose the heart. We cut the pericardium up to the base of the heart. Now we pin the pericardium through the base of the heart to the board. When we fix the base, the apex of the heart is pulled downwards with each contraction. This ensures that the force of contraction is directly transmitted to the lever. This increases the amplitude of the recording. We hook a bent pin through the apex of the heart. We ensure that the pin does not rupture the ventricle of the heart. We tie a thread to the end of the pin and tie the other end of that thread to the starling heart lever. The thread must be kept vertical and taut so that the contractions of the heart can be transmitted to the lever. The writing arm of the heart lever is kept horizontal and the writing point is just touching the drum of the kymograph. The drum is rotated at a slow speed. The contractions of the heart are recorded on the drum as a cardiogram. We first study an ideal cardiogram. This cardiogram is difficult to obtain using standard laboratory equipment. The ideal cardiogram shows three waves. The waves represent the activities of the different chambers of the heart. The downgoing wave represents systole or contraction, and the upgoing wave represents diastole or relaxation of the heart chamber. In the ideal cardiogram, we see the S wave, which represents contraction and relaxation of the sinus venosus, the A wave, which shows the atrial contraction and relaxation, and the V wave, which indicates ventricular contraction and relaxation. In a standard lab, we usually obtain a cardiogram which can show us the cardiac activities in the atria and ventricles. The contraction of the sinus venosus is not strong enough to register on a standard cardiogram. In this standard cardiogram, we can see wave A, which shows the contraction and relaxation of the atria, and wave V, which shows us the contraction and relaxation of ventricles. The wave S, representing sinus venosus, is not seen. We learn about some important properties of the heart through this experiment. You can see that the heart beats automatically. It generates its own rhythm and beats on its own. This is called the automaticity of the heart. The heart beats in a fixed rhythm. After every A wave is the V wave, and after every V wave is the next A wave. This is called rhythmicity of the heart. The waves on the graph show us that the chambers of the heart contract. This is called contractility of the heart. We will observe more experiments through which we will discover more and more properties of the heart.